Hello, my name is Kara Falvey, and today I will be presenting on the Conus magus, or the magician's cone snail. The objectives for today's presentation is to give you a brief introduction to the cone snail, including the habitat and life cycle. After, I will be discussing the current research and social implications of the research in today's world. The Conus magus is a species of sea snail, a marine gastropod mollusk in the family Conidae. They are a tropical species, and they are mainly distributed in the western central Pacific in tropical regions between 40 degrees north and 40 degrees south of the equator. The area shaded in red on the map is the most common distribution of cone snails. They live in the intertidal regions in sandy coral reefs and shallow bays. Conus magus, like other members of the mollusks, has a soft body and protective shell made of calcium carbonate. This shell serves as protection from predators, such as crabs who use their claws to pry open its shell. The unique cone shape of the shell makes it harder for the crab to hold and manipulate the shell, giving the snail more chance of being disregarded by the crab and surviving. The shell shape is also helpful for boring into the ocean floor, allowing the snail to hide from predators. Cone snails live a mainly solitary life. However, they are not hermaphrodites like most snails. They mate once per year, and the females lay several capsules with varying number of eggs, which you can see in the picture. Most will not survive the first few days of life. If they grow into mature adults, their lifespan is about 10 to 15 years. Like others in the Conidae family, the Conus magus is carnivorous and predatory. Because the snails are slow-moving, they capture their prey, which is mostly small fish, by using a radula tooth to inject toxic venom, which you can see in the picture. This venom is one of the most powerful venoms found in nature, and the toxin is called conotoxin or conopeptides and immediately paralyzes their prey so they can consume them. Injuries from cone snails to humans have been documented. These injuries occur mostly when they are stepped on or picked up. There have been some documented cases of fatalities in humans due to cone snail venom, but these are very rare. In the next slide, you will see a brief video of a cone snail attack that was featured on National Geographic.
The fish is paralyzed within seconds, reeled back in, and swallowed. With a full belly, the cone snail returns under the sediment, its siphon peeking out as a reminder and a warning. Heed it.
As seen in the video, the cone snail effectively utilizes its venom to survive in the wild. However, scientists became increasingly interested in this venom as well. It has been found that the Conus venom has compounds that act on specific ion channels or ionotropic receptors, which triggers other physiological functions. The natural capability of conotoxins to target these sites is being studied for use for potential drug development. Studies have suggested that an individual cone snail venom comprises greater than 1,000 pharmacological active components. The picture is a diagram of the mu conotoxins, which is one of the conotoxins that have been researched for clinical use. Mu conotoxins specifically target voltage-gated sodium channels. Hyperexcitability and mutations of sodium channels are responsible for perception and transmission of inflammatory and neuropathic pain states, which means that they have potential therapeutic use as analgesic compounds in the clinical management of pain conditions. So the question is, why do we care? Well, an estimated 125 million Americans are suffering from either acute or chronic pain. In the mid to late 1990s, the American Pain Society recognized pain as the fifth vital sign. This allowed for medical providers to increase availability of prescription opioids for chronic, non-cancer pain. The emphasis at the time was the patient's right to the assessment and management of pain. Unfortunately, as they became more liberal to prescribing opioid medication, the misuse of these medications also increased. As you can see from the graph, the number of deaths related to prescription opioids, noted in blue, has steadily increased since 2000. It is estimated that about 4% of the adult U.S. population misuses prescription opioids. However, the most visible manifestation of the opioid epidemic is the rising number of overdose deaths, estimated at more than 33,000 in 2015. The epidemic is such a concern in the United States that the American Medical Association has removed pain as the fifth vital sign. Therefore, it will be helpful to find a long-term solution for controlling and ultimately eradicating this epidemic. This may mean developing alternative, non-addictive methods of pain relief. The omega conotoxins are the most widely studied conotoxin for medical use, and these were initially isolated from our snail, the Conus magus. They act on calcium channels, which impact neurons that terminate in the dorsal horn of the spinal cord. Upon blockage of these calcium channels in the dorsal horn by the omega conotoxins, neurotransmitter release is inhibited and the sensation of painful stimuli is prevented. The omega conotoxins have been studied for the treatment of neuropathic pain, or pain that develops specifically due to nervous system damage or dysfunction. Neuropathic pain can often be a range of prior conditions, including infection, trauma, autoimmune diseases, metabolic disease, vascular disease, and cancer. These conotoxins were developed as an analgesic for chronic pain under the generic name Ziconotide by Alnylam Pharmaceuticals and underwent extensive animal and human trials. Ziconotide was approved by the Food and Drug Administration in 2004 for use. While the development of Ziconotide is a breakthrough for the medical community, like all medications, it has both its pros and cons. Ziconotide does not cause tolerance, dependence, or respiratory depression, which is seen with most opioid pain relievers. And because of the receptors it works on, it also does not have adverse effects that are primarily involved with the central nervous system, such as dizziness, confusion, and headache. And according to one study, patients reported significant improvement in their pain, which ultimately is the purpose of the development of this drug. However, Ziconotide can only be administered through an invasive intrathecal infusion pump, which is an injection into the spinal canal. The dosing must also be very controlled, as too high a titration was accompanied by side effects including sedation, dizziness, and hallucination. As expected, slower titration of the drug provides a more modest effect. Finally, due to the potential side effects of hallucinations, the drug is currently contraindicated for use in patients with a history of psychosis. As noted in the previous slide, Ziconotide works on certain receptors which are not linked to the central nervous system. While this eliminates the negative side effects with crossing the blood-brain barrier, it also limits the administration of the drug. New research conducted is attempting to find ways that this omega conotoxin can be more accessible to a wider population. In previous studies conducted, it was seen that a peptide known as the transactivator of transcription domain, or TAT, had been shown to have an ability to penetrate the blood-brain barrier. Therefore, scientists utilized this information with the omega conotoxin to make modifications to the existing drug Ziconotide. The study conducted in mice showed that when modified, the drug was able to penetrate the blood-brain barrier and have a pain-reducing effect when administered both intravenously through a vein or intranasally through the nose. The study found that there was a need for a higher dose of the drug to achieve the effect. However, the analgesic effect lasted for a longer period of time. In the last few decades, more and more studies have emerged in the pharmaceutical development from animal products. These have been derived mainly from venoms, toxins, and secretions from animals. The cone snail is just one of many animals that have been productive in medication discovery and scientific research. The hope is that there are many new potential therapies on the horizon and that this specific conotoxin can help reduce the dependence on addictive medications while still providing pain relief that will help improve the quality of life for those who are struggling with chronic pain. I hope that you enjoyed learning about the magician's cone snail and the wonderful contribution that they have made in the medical community.